Hi all, welcome back. This is the second part of Wednesday's lecture. I'm making two separate parts so it can be easy to it's easy to upload. We're still talking about DNA damage and repair, and there will be another lecture on DNA damage and repair on Friday. All right. So in the previous set of slides, we were talking about focusing more on chapter 10, 326 to 338 pages. In the last set of slides, we were talking about DNA damage repair in E. coli and how these different proteins, different enzymes work together to repair the DNA damage. I also mentioned that the damage repair is quite similar in eukaryotes, but not exactly the same, and it's not very well understood either. So these are the enzymes that we talked about in the previous set of slides. So there's a mistake on that slide, so please don't use that. Use this one instead. And what the mistake was I added MLH to mismatch detection in that slide. That's not correct. The orthologous protein to mute S in eukaryote is MSH, and orthologous protein to mute L in eukaryotes is MLH and PMS. These work with BCNA and RFC, and we will talk about those later. Orthologous to mute H is possibly an exo1. We don't know. Now we don't know about a helicase. We don't know much about an exonuclease, but possibly an exo1. DNA pol lambda and ligase help fix the break. DNA pol lambda is the major and part of polymerase, of the core polymerase in eukaryotes. So the mechanisms, what we talked about in E. coli, and let me just talk about that one more time. In E. coli, if there is a mismatch, this is the parental strand that is methylated. This is a daughter strand, newly synthesized daughter strand or newly undersynthesized daughter cell that is not yet methylated. This mismatch will recruit mute S proteins. These mute S proteins will go and bind where the mismatch has happened. And this mute S, the binding of mute S, it will scan and it will find the correct position. It will recruit mute L. This mute two of these dimer of this mute L will bind together. This binding of this mute L to mute S will activate mute H, which is an endonuclease, and this mute H will then make the break on daughter strand. Mute U or UVRD is an helicase that will open the DNA molecule over there at the repair part. And exo1, which is a 3 to 5 prime exonuclease, or exo7 or 8, or also known as RECJ, will act as a 5 to 3 prime exonuclease. Either of these will work. It depends on how this complex is bound. Pol3 and ligase will then fill the gap, the neck, and it's corrected here. In humans is a little different. In humans, the repair is continuously going on as the replication is occurring as well. It occurs mostly on this continuous repair is going on the lagging strand, because there's multiple break sites there. The mute S equivalent or orthologous to mute S in humans or in eukaryotes is MSH2. This MSH2 binds to the DNA molecule, and GTBP or B160 is another component of this MSH2 recruitment protein. These two bind, and the binding of these two DNA will bring MLH1 and PMS2. These MLH and PMS2 are mute L equivalent. These will activate the endonuclease. What's the helicase? We don't know. What happens next? We don't know very much. What exonuclease might be. Pol, alpha, delta, or epsilon. These are the units of the core polymerase, as you might recall. These will then fix the DNA, the error, and and will fill the gap as well. And so delta and epsilon are involved in DNA damage or in exonuclease activity, and alpha is a polymerase, so this polymerase will then fill the gaps. So this is what happens in eukaryotes or in human. As I mentioned, this MSH interacts with sliding clamp, which is called as BCNA in eukaryotes. This forms the component of repliosome. That means all the proteins that are present at the replication fork and working on the replication of DNA. These are usually recruited to the site of discontinuous synthesis on the lagging strand because the error happens more commonly on lagging strand because of the multiple Okazaki fragments or smaller portion DNA synthesis. This interaction with sliding clamp also recruits these mismatch repair protein to 3' end of the leading strand. So both strands will have the DNA damage repair, but it's lagging strand that's where it's always being a part of the repliosome. MutAl, MutAl alpha, which is MLH1 PMS2, is a latent endonuclease. That means it's inactive. Usually it's inactive. It gets activated when there's a mismatch. There is a MutS already present on the DNA strand, the proliferating cell nuclear antigen or PCNA, and replication factor C. All these are present. This is an ATP dependent reaction, so ATP is required for MutAl to activate as an endonuclease. So RFC, this replication factor C, this appears to be present only for endonuclease activation. It's only involved in PCNA loading. So PCNA here, this is PCNA. This teal and brown circle. So this is your beta clamp. What we talked about beta clamp when we were talking about DNA replication. So this PCNA interacts with MLH1 and PMS2 to activate MutAl, which will then be activated as an endonuclease and make a cut. This PCNA is very important, and as I mentioned, it's a DNA clamp. It acts as a processivity factor for DNA polymerase, delta, and eukaryotic cells, and it is essential for replication as well as for repair. PCNA is a heterotrimer, like we talked already. It's how the whole enzyme is a trimer of all different components, one on the leading strand and two on the lagging strand. So this PCNA or these beta clamps are also trimer, one on the leading strand and two on the lagging strand. Excuse me. Yes.
So, you were watching a movie, and you know what?
Yes.
After the movie was done, I was talking to Dad about a different movie that I have seen. And it was almost the same as that one movie we were watching.
Okay. Can I work now?
Mama, no. E.T. was an alien.
Oh, yes.
And you know who my favorite was?
Who?
E.T.
Okay.
Bye.
Okay, that was my daughter. Okay, back to PCNA and how this all works. So PCNA is a homotrimer. It's achieved processivity by encircling the DNA because it keeps everything together, keeps the alpha subunit, which is the polymerase subunit, close to DNA that way. It also acts as a scaffold. This recruits different proteins, and these different proteins that we just talked about in the previous slide, the MLH1 and PMS2 MutAl. These are all recruited at this scaffold of PCNA. This PCNA is very high levels of PCNA are found in cancer cells. So it's excuse me. So this PCNA appears to have an essential role in the cancer cells because they are continuously replicating and repairing. So it's fair to imagine or fair to think that this will be very important component in cancer cells. So a number of different labs are using small molecules that will inhibit PCNA activity. So that way, this can be a therapy molecule. So here in this example, you can see this is PCNA and RFCs. They work together. This is the replication factor. As a replication factor and PCNA, these bind to DNA. They hold the DNA because it's a clamp. It's important for DNA replication and repair. But if we inhibit this PCNA and it cannot bind to DNA, that will lead to DNA damage. Finally, it will lead to cell death. So same here, it will inhibit the DNA replication, inhibit the cell growth by inhibiting replication. DNA replication, the replication fork will stall and collapse, and that will lead to more DNA damage and cell death. So these appear to be pretty good candidate for cancer treatment. And this paper here, this is the PDF link for the same paper. This is a really nice overview of this mechanism. Okay. So that we were talking more about the replicative errors in DNA and how to repair those. Now DNA damage can happen by multiple different ways, and not always just the replication error, because replication errors are quite infrequent. The physical and chemical damage can happen to DNA, and there are different molecules, different chemicals, different compounds can do that. Physical damage by excessive heat, by UV radiations can change or damage the DNA as well. Damaged DNA is not mutated yet because this damage can be repaired, but if the damage is too much and it cannot be repaired, or the repair system doesn't catch it in time, then that leads to mutations. Chemicals or conditions that damage DNA are called mutagens or mutagenic substances. Damage can alter base pairing, it can interfere with cellular machinery, so polymerases or transcription or replication. It can physically break the sugar phosphate backbone. Clastogens usually do that. They break the DNA strands. Benzene is a good example of a clastogen. DNA can also undergo spontaneous damage due to action of water, because our cells have a lot of water. Usually, many reactions release water molecules, and these can damage DNA as well. DNA damage from spontaneous hydrolysis and deamination is quite common. Deamination is very, very frequent, the most frequent type of DNA damage, which occurs spontaneously in the cell. So the cells are well aware of this problem, and they have a system in place to remove this or fix this sort of damage. We already talked about this, that cytosine, when it gets deaminated, it generates uracil, which is unnatural for DNA. U pairs with A instead of G that the cytosine will pair with. So here, the deamination of cytosine leads to formation of uracil, and that's one of the reasons why the DNA has thymine instead of uracil, because spontaneous deamination of cytosine will result in uracil, and if uracil was the base pair that the DNA uses, then it will be hard for the cell to tell whether it's a uracil or it's a uracil. That's because of the cytosine deamination. Adenine and guanine can also go through spontaneous deamination. Deamination of adenine leads to formation of hypoxanthine, and this hypoxanthine pairs with cytosine instead of thymine, so that will then lead to cytosine guanine. Guanine deamination leads to xanthine, but xanthine also binds with cytosine just like guanine, so there is no base pair change. Depurination is another way of spontaneous hydrolysis and deamination. Depurination is another way of DNA damage. What happens in depurination is, this example is right here. So remember, your nucleotide has three components. So there's a sugar, there's phosphate, and then there's base. If the base is released and only sugar and phosphate is left there, that's the depurination that results in abasic sites. There is the deoxyribose phosphate is lacking a base. Depurination or formation of abasic site will halt transcription, because transcription cannot occur if there is a base pair missing. There will not be, it will stop there. Most other mutations will not affect further replication or transcription, but there will be an effect on what protein is formed. And we already talked about this as well. The deamination of 5-methylcytosine generates thymine. These are both pyrimidines, cytosine and thymine, so it's usually slips from a repair mechanism, and following a round of replication it will be fixed. Methylated cytosines are hotspots for spontaneous mutation in vertebrate DNA, and we talked about this, that these methylcytosines are quite common in invertebrate genomes because these make the CPG islands, or they make the CPG sites. These CPG sites, because these are a lot of these CPG sites are present in promoter regions, whether methylated or not, but about 80% of CPG sites are methylated. So there are a lot of cytosines in methylated cytosines in human genome or in vertebrate genome in general. And in vertebrate genomes, it's a little different because there is not these invertebrate genomes are not heavily methylated. In a way to study the methylation, cytosine methylation, we use bisulfite sequencing, and we already talked about bisulfite sequencing when we were talking about DNA methylation and epigenetics. What happens in bisulfite sequencing is when you treat DNA in vitro with bisulfite, it converts cytosine to uracil, but it will not convert methylcytosine. Methylcytosine will stay as such, and that way we can know which cytosines are methylated and which ones aren't. This is an example here. So we have these, this is a methylated allele, this is unmethylated allele from two different organisms, or probably of two different populations. You treat with bisulfite, methylated cytosine will still stay cytosine and everything else will be uracil. And when you do PCR and sequencing, then you looked at these sequences. The sequences will be cytosine here in this allele, but no cytosine in this one, which suggests that this was methylated, and this site was methylated in this allele. DNA damage can be caused by alkylation, oxidation, and radiation. In alkylation, there is you can see here, a ethyl or methyl group are transferred to reactive sites on the bases. Most of this can be attached to both. Ethyl group can be attached to either an N or an O. Now, methyl group will mostly attach to an N. The most common alkylating agent is ethyl methane sulfonate, or EMS in short. This is used in lab, or used to be used heavily for causing experimentally random mutations. So that way you take a lot of individuals, a lot of organisms, and treat them with EMS. Some of this, because this mutation is random, some of them will have some genes affected or mutated, some will have other genes affected. And then you look for phenotype to see which genes, what phenotype is associated with what gene, what was mutated in these individuals, and you can put then gene to function, assign a function to gene. This used to be quite common, these random mutations, and these still are quite common, but we can do more mutation experiment or functional genomic experiments in other ways now, for example CRISPR or RNAi or transgenic individuals. Addition of methyl to G makes it methylguanine, and it pairs with the T and changes a GC to AT. I will put a little bit more here. This is the deamination. So that if this amine group is removed, this will lead to deamination of guanine here. If you add a methyl group to guanine, that will, so say you add a methyl group here, this methyl group, addition of this methyl group will make it methyl guanine, and which will pair then with thiamine. Alkylation can happen at either at O or an N, and oxidation. This oxidation can be due to say UV light. All these different ways the DNA can be damaged. So we will talk. This is the first part of our DNA damage and repair chapter. We will talk more about DNA damage and repair, more focusing on repair in the later part, and we will talk about how the cell repairs these damages, and we will go in detail with two major pathways. That's how the DNA damage is repaired: non-homologous end joining and homology-directed repair. We will not talk too much in context with CRISPR, but more how the cell by itself in situ will repair those sort of double-stranded or single-stranded breaks. Okay, and this is all for now.